Hey Bio 30s, it's Manchak again with your final lesson for concepts C4 and C5. So at this point, if you're watching this, you should have already finished up concept C4 video, which introduced you to Mendelian genetics, the laws of dominance, segregation, independent assortment. We had a look at some terms like allele, heterozygous, homozygous, dominant, recessive, those kinds of things. Then you should have proceeded on to the first video of concept C5, which had you looking at simple monohybrid crosses to start to predict probabilities of certain phenotypes or genotypes in offspring. And that also went into looking at the pedigree as a tool to complete a family tree of a certain trait or disorder. I hope at this point you've taken some time to work with those Punnett squares and work your way through the worksheet that I assigned for you, worksheet number 1, which was Punnett squares and probabilities. And if you haven't had a chance to do that yet, please pause right now, go back and do that, because I'm going to be going into some more complicated patterns of inheritance that we're going to discuss in this concept, or this part of the concept. And so to wrap up C5, I need you to have that first part of C5 done first. So if you've done all of that, then we can continue on. So this is our second video in C5, and we're going to be looking at other patterns of inheritance, which includes situations of multiple alleles, incomplete dominance, codominance, and then the probabilities of the dihybrid crosses, and we'll talk about what a dihybrid cross is. We're still working through this range of textbook pages, and we're going to have a series of videos associated with this. So we'll start out with this one on multiple alleles, and I'll mention when you can get it to that one, and then some incomplete dominance, codominance, polygenic traits, and epistasis. These last two concepts we'll talk about at another point, so don't stress about them too much. And then we'll talk about dihybrid and two trait crosses. All of these are Amoeba Sisters videos. Again, I've said their stuff is fantastic, and I think that that you should definitely give them a watch. I know it's a lot of videos. I know you are investing a lot in these two concepts, but the rest of Unit C, the concepts are relatively short and relatively straightforward. So I apologize for the length of what's going on here, but we'll continue to work through all of this and make our way through it with no problems. There are going to be 3 more worksheets associated with this at the end of this concept. They're actually not too bad. They won't take you too long to do, but I really think they're worthwhile jumping through and working your way through. And you can find those on the Google Classroom under Notes and Worksheets for Unit C, as well as the keys are under Resources and then Worksheet keys. So I guess without any further discussion, I'm going to jump ahead into this section of concept C5, which is other patterns of inheritance. And in particular, what we're going to be talking about, we should have a pretty good handle of the terms dominant, recessive, genotype, phenotype. You should be able to do what we call a monohybrid cross, and that's just looking at one trait in an individual. And those traits can either be dominant or recessive in their phenotype and in their genotype. What we're going to do now is we're going to add in some other forms of inheritance that make things a little bit trickier, but really not too difficult to work with once you've got a grasp on what you're looking at. So the first pattern of inheritance that's a little different from monohybrids is what we call a multiple allele situation. And just off the top, basically what we're referring to here is that instead of one trait having 2 possibilities, let's use our pea plants for example. In the monohybrid situation, there was 2 possibilities for the shape of the seed. It was either round or wrinkled. And those were the 2 alleles that we had associated. Sometimes, in certain cases, we have a trait that has more than 2 alleles associated with it. It could have 3, 4, 5, 6 alleles. Each of them has a different physical expression of that trait. So we call this kind of situation a multiple allele situation. And one of the things that we do when we're working with introductory genetics like this and trying to understand all of these multiple allele situations or different ways that we can present inheritance, we often work with organisms that show us some of these patterns in a very simple way. So as Gregor Mendel worked with pea plants and discovered his laws, we can work with other organisms, and one of them is a Drosophila, which is a fruit fly. So Drosophila melanogaster is fruit flies, the fruit flies that you find flying around your house, just the little tiny ones that are a little bit annoying but don't really do anything. Those are what we're working with here. When I was in university, we used fruit flies quite frequently as we were working through our continuation of what we're studying now with respect to genes and heredity and alleles and all of that kind of stuff. So classical genetics. And we would actually do labs where we would take a bunch of fruit flies and we would literally hit them with chloroform. We'd put them in a jar, put a drop of chloroform in the top, and what it would do is it'd stop them from moving. Wouldn't kill them, it just stopped them from moving. And then we would dump those little critters out onto a petri dish and get them up underneath a dissecting microscope, and we would start to classify them based on whatever trait we were looking at. And it could have been something like wing shape or eye color or body color, and we would have been grouping them. And it was actually kind of funny. I do remember my mom used to work in the same building that we did these labs in, and she always knew when the Bio 108 students, or 107 students, pardon me, were doing the Drosophila lab, because there would be fruit flies all over the building at that point. Because what happens is if you're not quick enough to sort all of your little Drosophila, they start to come back awake again and fly around. And so you've got little Drosophila flying everywhere. You've lost your sample. It's a bit of a mess. So you have to learn to work quickly, is what I'm saying. So why we use Drosophila is that they tend to show traits that are pretty simple to look at. They're big enough that we can find these traits, and we can breed a whole bunch of them, a whole bunch of generations very, very quickly if we want to. So Drosophila melanogaster, these fruit flies, have 4 different eye colors that are easily observable under a dissecting microscope. The eye colors are red, apricot, honey, or white, or apricot. Sorry, I always say apricot, and I don't know why. Each fly carries only 2 genes for eye color, okay, or alleles. The combination of these 2 determines the eye color for the organism. So these 2 possibilities are wild type, which is the most common type of gene with multiple alleles. So in the case of the Drosophila, wild type is red eyes. And then there are mutant alleles of this gene, which are colors other than wild type, which includes apricot, honey, or white in the species of Drosophila we're talking about. What we're working with here then is you could be a normal Drosophila with wild type or the most common allele, which is red eyes, or you could be a mutant of some sort with an apricot, honey, or white eye. And we would call it a mutant allele. Where we're heading next here then is what we have here as a multiple allele situation. And in these multiple allele situations, what we need to do to figure out some probabilities and predictions of genotypic and phenotypic ratios, we need to sort out what's called a dominance hierarchy. So a hierarchy is a ranking system. You know, this is above this is above this, right? We create a ranking system based on what is dominant, what is the most prevalent form of the allele, and then what are the other ones that or the trait, pardon me, the most prevalent allele for the trait, and then what are the other possibilities associated with that. By the way, these are fruit flies right here, and you can quite easily see the difference in the colors of eyes, and there's other things that you could pick out, and we'll talk about in a little bit. I'm going to move my video up here just so that we can talk about what's down below. So what this is representing here is a dominance hierarchy. we know that the most common allele and expression of allele is for the red or wild type eye color. So that's this one right here, the red eyes. But then we also know that there are mutant alleles, so apricot, honey, and white, that are associated. And if you were to actually take a sample of, let's say, 500 of these Drosophila and sort them out, you could probably get a pretty good sense as to what's wild type and what's mutant. And you could probably even get a better sense as to the dominance hierarchy. And the way to think about it is that the more individuals you have with a particular phenotype, a particular expression of a trait, the higher up that is on the hierarchy. Because if you have, and we'll look at it this way, if you have the E1 allele, so this is E for eye color, 1 representing the wild type, 2 is going to represent apricot, 3 is honey, 4 is white. Notice how we're doing a slightly different way to organize this cross, or this set of alleles. Instead of just doing big uppercase and lowercase, we're going to do E and then a superscript 1, 2, or a 3. So with E1 for the wild type, you'll notice that all the possibilities with E are these right here. So you could have 2 E1s, you could have E1 and E2, an E1 and E3, or an E1 and an E4. And what happens, and what this is telling us, is that because this is the top of our dominance hierarchy, it is dominant over apricot, honey, and white. So what that means is if there is an E1 present, we are going to have an individual with red eyes, or the wild type eye color. And what we would do then is if we had 1,000 individuals, and I'm going to throw a number out, and I don't think it's perfectly accurate, but if we had 1,000 individuals and we sorted them all out, we would find that the majority, let's say 500 to 600 of those individuals, would have the wild type eye color. Okay, and then as we move down the list, we have the less dominant alleles. Now we've moved into the mutant alleles here, but there's a hierarchy here. And what it's saying is that the E2 allele is dominant over honey and white. So if you have an E2 allele in one of these 3 combinations, the individual will have apricot eyes. And then apricot's dominant to honey, but honey is dominant to white. So the only possibility of getting a white-eyed individual is if you have 2 of the E4 alleles. You've got E4 alleles in each of these cases here, but it's being overruled by one of the other alleles in that case. So I hope that makes a little bit of sense. So when we have a multiple allele situation, like we do for eye color in Drosophila melanogaster, there is one trait, one allele that rises to the top where the individuals are most commonly found to have that particular physical expression, and then a hierarchy is created with that one on the top and then numbers dwindling further down. So if we said maybe we have 600 individuals here, that we might have, I don't know, let's say 200 individuals here, so 600 and 200 is 800. You might have 150 individuals here and only 50 individuals here. And that creates our dominance hierarchy. So based on what we just saw, what would the phenotypic ratio of the offspring from mating the following Drosophila individuals be? So I've set up a little bit of a Punnett square for us. I'm going to write in white to start with. And here's our cross. And we're going to set these up similarly to what we did before. But notice now that we're not using the capital letter and the lowercase letter, the big E and the little e. We're using these superscript numbers to indicate the eye type that they have. So this is a wild type eye color. That's their phenotype. But we notice that this individual is heterozygous, meaning that they have 2 possibilities. So I'm going to write them up here as E1, and we'll split that up due to segregation, and E4. Okay, and then this individual has apricot eye color because 2 overrules 3, just like 1 overrules 4. And we're going to set this up over on this side as E2 and then as E3. Okay, so then we've set up our Punnett square, and the whole thing about the question is that it's asking us to determine the phenotypic ratio. Okay, so we're going to start to put these together. E1 pairs with E2. Always list the more dominant one first, as we have always done so far. So this individual will turn out to be E1, E2. Then we'll end up with an E2 and an E4. Okay, then we end up with an E1 and an E3. Pardon me for the horrible writing. And then finally we end up with an E3 and an E4. So if we're trying to sort out a phenotypic ratio, so phenotype, remember, is physical expression, then what we're ending up with in this situation with this phenotypic expression is that we've got two individuals with E1, which overrules everything else. So we're going to have two with red eyes, okay, two, one with apricot eyes, A-P-R, apricot, okay, horrible writing, two, one individual, which is interesting, we've got somebody new coming out with the, why am I blanking on it, with the, oh, it's all disappeared, with the honey-colored eyes, of course, because I switched slides. So we had a two red to one apricot to one honey for our phenotypic ratio here. Okay, remembering that certain things overrule something else based on a dominance hierarchy. So if ever you hear about dominance hierarchy, you should be thinking about a multiple allele situation. If you need to go back, if you're filling in your notes, go back and just really quickly rewind it so you can get the last little bit before I erased it there. But I hope that makes sense for multiple alleles. Okay, so that's our first new type of inheritance, pattern of inheritance. The next one we're going to talk about is incomplete dominance. Okay, so again, we're going to play around with something new. We're doing simple Punnett squares right now. We're not doing anything more complicated than that. And in this case now, we have a situation where, you know, in the last case, the multiple alleles, we have for one trait, which is eye color, we had multiple possibilities for that eye color. In the same case is true in incomplete dominance, is we would expect if it was a regular this or this dichotomy, you know, you would have white eyes or you'd have red eyes, right? We would expect that those would be the two options. In incomplete dominance, there's a third option available, and that third option results from the two alleles being equally dominant in a heterozygous situation. So if we had one allele from one scenario or one allele from, let's try that again. If we had one allele of the one type and we had the other allele paired together in a heterozygous situation, then they are equally dominant in an incomplete dominance situation. So this results in the expression of both forms of the allele in a heterozygous individual in the cells of that organism, producing what we call an intermediate phenotype. Okay, that intermediate phenotype is I like to think of it as more or less a mixture of the two. So the best example I can give you right now to play around with this is snapdragons, which is a kind of flower you may have found in your garden before, and they're incompletely dominant for flower color. And so what that means is normally snapdragons would have red flowers and white flowers. Okay, and if you have an individual who is homozygous for white, they will have white flowers. If they're homozygous for red, they will have red flowers. If they're heterozygous, they will have a mixture of the two, and they would have a mixture of red and white, and a mixture of red and white is pink. So pink is the intermediate, which we would find in individuals who are heterozygous. So here's an example of how we would work with this sort of situation. So we've got we've got a homozygous red snapdragon crossed with a homozygous white snapdragon. What are the individuals that we would expect to get in this cross? So this is the ignore that, that was awful. This is the sort of the way we express, or the notation that we use for this. So again, we're going to use a big letter like C to represent color in this case, and then we're going to use a superscript here for R. In this case is going to represent red. So this is C superscript R, C superscript R. That's our first individual being crossed with C superscript W. C superscript W. So this is our cross. So we can set up a Punnett square the way we would normally, and you know what? I'm going to cheat here because we only really have one type of allele coming from each of the parents in this cross. We have a CR allele coming from this individual and a CW allele coming from this individual. And so what this allows us to do then is determine what our genotypes are going to be for the offspring of this cross. And you'll see that all of the offspring are going to come out to be CRCW. Now, since this is an incomplete dominance situation, where when we have a heterozygous individual, both of the alleles are expressed equally, so we end up with a mixture or an intermediate of the two. That means that based on this cross, 100% of the individuals will end up having that pink color when they're done in their flowers. Okay? Now the follow-up question here is, what would happen if you crossed the F1 generation? This is the F1 generation here, created from that first cross. And so what the second part of this question is asking us to do is basically take two of these individuals, two of the pink colored plants that were produced from the first cross, and cross those individuals. Okay, so that is our cross there. So I'm going to have to fill this one out in full because we're going to end up with some interesting results here. Okay, so I've split these up according to segregation, our law of segregation, and I am going to fill in what we have. So this individual here will be CR, CR. This individual here will be CR, CW. Okay, this individual here will also be CR, CW, and I like to just be consistent. If I put CR, CW here, I'll put the R first in this next one. And then CW, CW again. And so we end up with some interesting phenotypic ratios when we're done here, because that's what the question's asking us for. So we're going to have one red individual for every two pink individuals, right? Because there's two pink individuals, one here and one here, for every one white individual. Okay? What I wanted to mention at this point is when we get into these different patterns of inheritance, like incomplete dominance, multiple alleles, or codominance, which we're going to see in a second, the ratios that you got comfortable with those monohybrid crosses at the beginning of C5, they're thrown out the window a little bit. And that's how we came across these ideas of other patterns of inheritance, multiple alleles, incomplete dominance, codominance, is those ratios just don't seem to work anymore. When you cross two heterozygous individuals previous to what we've just talked about, we should have ended up with a phenotypic ratio of 3 to 1. 3 to the dominant, 1 to the recessive. We don't have that here. We have a 1 to 2 to 1 ratio. And that ratio does, all of a sudden we end up with another phenotype, but we also end up with a ratio that just doesn't seem to jive. That's what gave us the evidence that there are other patterns of inheritance out there. So the last one we're going to discuss in this section is something called codominance. And I'm going to give the biggest example, I think, for you guys here is to have a look at this one. And in the one of the videos goes into talking about multiple alleles and blood types and how they work. So check that one out. And then there's also that second video on incomplete dominance and codominance that you guys should check out as well. If you guys see me looking over here, my other monitor's over here, and I've got, you know, some notes that I'm kind of reading off of to remind myself of what I want to talk about. So codominance is the last type I want to talk about, well, before we get to dihybrid crosses. And this is a similar situation to incomplete dominance, but in incomplete dominance, what happened is we had when an individual was heterozygous, we had a mixing of the two. So red and white made pink. Okay. In codominance, what ends up happening is they're also, when we have a heterozygous individual, both traits get displayed equally. Okay, they're fully visible. That phenotype is fully visible if for both traits or both alleles, if the individual is heterozygous. Okay, so when the contribution of both alleles are fully visible in the phenotype, we encounter codominance. And so some examples of what this might look like, and this is probably the biggest one. We studied blood types last year in Bio 20, and we studied the ABO blood types. So you could have type A blood, you could have type B blood, you could have type O blood, or you could have type AB blood. Okay, and we talked about what the A and the B and the O mean. The A means you have the A antigen on your blood cells that identifies you as having type A blood. If you have type B blood, you have the B antigen on your blood cells only, and it identifies you as having type B blood. If you have type O blood, you have no antigens of those ABO types on your blood cells. And then if you have type AB blood, you have both antigens. Okay. Where this comes from is codominance, is that if an individual ends up heterozygous, in other words, they have, and the I is just what we use to represent the blood in this case. If we have an individual that has both IA, so the A antigens and the B antigens, they have the type AB blood. And so they will have both antigens present on their red blood cells. Okay, so that's an example of that. Sickle cell anemia, which is a component of, in sickle cell anemia, a component of hemoglobin called beta globin, produces three phenotypes. It produces HB for hemoglobin, this possibility, which is homozygous for A, this possibility, which is heterozygous, and this possibility, which is homozygous for S. The first individual is unaffected by sickle cell anemia. Okay, so they'll be normal, like most of us, like you and I. I mean, I'm as normal as I can be. Not that normal. And then the second individual will have a mild version of anemia, and the third individual will have full-blown anemia. So what's happening with that mild version is that some of the A is being expressed, making some of the cells normal, and some of the B is being, or the S is being expressed, making that a little bit abnormal. Okay, A for sickle, or S for sickle. And so this individual expresses a little bit of both. And the third individual right here has full-blown sickle cell anemia. Sickle cell anemia means your red blood cells aren't shaped properly, and it makes it difficult for them to pick up oxygen if they're improperly shaped. Their surface area isn't quite right. They don't maximally grab onto the oxygen molecules the way they should. Another example, a bit of a farming example, is with shorthorn cattle, where we're talking about hair color in this case. There are red shorthorn cattle, there are white shorthorn cattle, and then there's a mixture where they appear kind of frosty with a mixture of red and the frostiness of the white. And so we work with these codominance situations the same way that we work with what we did with incomplete dominance. And I just noticed there's a mistake on here. This should be a W for each of these, so as not to cause confusion, because it says a red shorthorn with a white shorthorn. I think you guys can sort of figure this stuff out already, so I'm going to just go really quickly through it. There's our white individuals gamete. There's our red individuals gamete, and you guys should see at this point then that all of the offspring of this cross are going to end up with that frosted appearance where they have both red and white hair being exhibited at the same time. And then if we're talking about phenotypes of offspring for someone with homozygous type A blood being crossed with homozygous type B blood, again, we're talking about blood types here, and we'll use that I, that I to represent this. So here's our homozygous type A blood, and they would be IA, right? I'm just going to focus in on this square. And this individual would be IB because they're homozygous for B blood. And so we're going to end up with a situation where all of their offspring will have type AB blood. Hey, that's pretty good because then those individuals are all going to be universal recipients. They can take anybody's kind of blood, if you remember back to Bio 20. Okay, so that's codominance and how that works. Okay, so we've talked about multiple alleles, we've talked about incomplete dominance, and we've talked about codominance as different patterns of inheritance. And we know those patterns exist because when we start to look for expected phenotypic ratios that we might have found with just simple dominant recessive sort of pattern of inheritance, things don't work out that way. And so we have examples out there of multiple alleles for one trait, or we've got an example of a heterozygous individual as in incomplete dominance, where if you have that individual, they show a mixture of the two alleles, or in codominance, you'll see the expression of both of those alleles in a heterozygous individual. In some cases, the interaction of genotype with the environment will also vary the phenotype or the expression of a trait. And an example I can give you of that is Himalayan rabbits have black fur when they're bred at low temperatures, but white fur when they're bred at high temperatures. Don't spend too much time worrying about that. Okay, if you wanted to pause now, there's some questions on page 612 you could jump into if you'd like. If not, I'm going to continue on. Ignore the case study. You can have a look at it on page 611 if you'd like to, about blood types and sorting that out. But what I'd like to get into now is finishing up concept C5, which is discussing dihybrid crosses and polygenic traits. Okay, if you want a break right now, go and watch the Amoeba Sisters video on dihybrid and two-trait crosses if you'd like. It's about 8.5 minutes long. It might give you a break from listening to me. If you don't want a break from listening to me, or you're back from looking at that video, then we're going to continue on. So a dihybrid cross is what we have when we're looking at two genes at once, and each of those genes or each of those traits has more than one allele. We're going to keep it relatively simple. We're not going to take, a trait that is just simply dominant and recessive, and then we're not going to take another trait then that is going to show incomplete dominance and try to figure stuff out. We're going to take two traits, and we're going to have a look at an individual with those two traits and cross them with an individual with those two traits and try to predict what's going to come up. And in those situations, all we're going to be dealing with is just a dominant recessive situation. So instead of a monohybrid, hybrid in that case meaning mono meaning one, hybrid meaning trait, right? Monohybrid meaning a one trait cross. We're going to do a dihybrid cross where we're going to do a two trait cross, and each of the traits is either going to be dominant or recessive. It'll have one set of alleles that will be dominant or recessive. Okay? These two crosses show Mendel's second law that each gene will assort independently, which leads to many possible gametes coming from an individual if we look at this. So we call this, remember, independent assortment. Remember that happens when we line everything up in metaphase 1 of meiosis 1, and we line them up based on mother and fathers being paired as a homologous pair, but you could have mothers on the left and fathers on the right, or you could have fathers on the left and mothers on the right, and whatever happens in that first pair is independent of how the second pair assorts itself or aligns itself. So this is a really complicated sort of diagram, but it's kind of showing this idea of independent assortment, and we're basically looking at what are the possibilities of the gametes that an individual could produce based on their genotype. Okay, and so if we look up here, the genotypes that we have, right? Both of these genotypes. So this is we're looking at our pea plants again. So just to set the stage a little bit, the big R and the little r are referring to round versus wrinkled. Okay, so this we'll call round and this we'll call wrinkled, right? You see the difference between the two. And then this Y, big Y and little y, are referring to the color. So, round is dominant over wrinkled, and I think they've got these mixed up. If I was to fix that, I would put these as wrinkled and round. I don't know what's going on in the key on here. But what we've got going on then is this idea of independent assortment, right? Where they're lining up and assorting themselves differently based on how their alleles are organized. But these individuals we would both call heterozygous for both traits, right? This individual is big R, little y, little y. This individual here is big R, big Y, little y. And when we create the gametes are going to have two alleles in them. One allele from the R and one allele from the Y. And so that's what we're showing over here. Is this individual over here can produce these 4 possible combinations for their gametes. They can produce a gamete with a little r, little y. They can produce a gamete with a big R and a big Y. They can produce a gamete with a little r and a big Y. And they can produce a gamete with a big R and a big Y. And this individual over here can do exactly the same thing. They have 4 possibilities for their gametes. Just to add something to this then, if we had an individual who had this genotype, let's look at that. Okay, so that's a big Y and a little y. This individual can only produce 2 possible gametes, right? They could produce a gamete that is big R, big Y, or they could produce a gamete that is big R, little y. I hope that makes sense. Okay, so then that individual can only produce 2 possibilities. If the individual was homozygous dominant for both alleles, they're only going to produce 1 possible gamete, which is big R, big Y. Okay, so I'm going to try to move on here because this is dragging on a little bit. So dihybrid crosses work in a similar way to monohybrid crosses. It's just that they get a little bit more complicated as we move through. Excuse me. So in a summer squash, and we're going to look at, we'll do this as a learning by example. In a summer squash, white fruit color is dominant over yellow fruit color. So if you have a capital W, your fruit is going to be white. If you have 2 lowercase w's, it's going to be yellow. And then we have disc-shaped fruit is being dominant over sphere-shaped fruit. So if you have a capital D, you are going to show a disc shape to your fruit. And if you have 2 little d's, you're going to show a sphere shape. So if a squash plant, true breeding for white and disc shape, true breeding means they are homozygous, is crossed with a plant, true breeding for yellow and sphere shaped. Again, true breeding means homozygous. So what this is, true breeding for white and disc shaped is big W, big D, big D. And true breeding for yellow and sphere shaped is little w, little d, little d. We can set up a really simple Punnett square for this. This individual here is only going to produce 1 kind of gamete. What kind of gamete is that? Well, they're only going to produce big W, big D. Okay? This individual here can only produce 1 type of gamete, little w, little d. Okay? I hope that makes sense. And so their offspring then are all going to have a big W and a little w, a big D and a little d. Sorry for the terrible writing. Okay? So all of their offspring are going to turn out like this. So we would expect for a genotypic ratio, 100% of them will be heterozygous for both traits, and 100% of the offspring are going to have white fruit that is disc-shaped. Okay? Now, moving on to this, this is where things get a little bit tricky. If we wanted to take offspring from the F1 generation, that was the individuals we put in here, and breed them, what would the resultant offspring look like? So here's the problem. Instead of each parent having one possibility for their alleles, like we saw in the first example, the F1 generation, each of those individuals has 4 possible arrangements, like we talked about on that slide with all the pea pods on it. These are the 4 possible arrangements. So we need to expand our Punnett square to accommodate all of those possibilities. Okay? And so we're going to fill this Punnett square in to sort of show how this is going to look like. And this is going to take me a little while because of this. So if we lay this Punnett square out like we normally would, the one individual can provide these possibilities for the gametes, and the other individual will provide the same possibilities for these gametes. Okay, little d, little w, big D, little w, little d. Okay, and then all of this is going to get brought together the same way. And so what we're going to do is we're going to bring these together as we would before. So this will be big W, big D, big W, big W, big D, big D. And then big W, big W, big little d. And then big W, little w, big D, big D. Sorry. Big W, little w, big D, little d. Okay, I hope that makes sense, and we're going to continue on doing that. So big W, big D, little d. Big W, little d, little d. Big W, little w, big D, little d. Big W, little d, little d. Okay, big W, little w, big D, big D. And I'm going to stop talking because this is annoying. So I will just start to ramble as I'm writing all of these down. And you guys can wonder, that looks like a B. Sorry about that. You can wonder about my sanity as much as you'd like. At this point, someday I'll figure out how to do this. I should have done this with a little video recording for you guys. But I didn't because, hey, why? That is supposed to be a little w, so I'm going to make this one a little bit bigger to show the difference. One of the problems with choosing things like w's is that it's often difficult to, you have to sort of exaggerate the size so you don't end up getting yourself confused. Okay, so there is our Punnett square. It takes a little while to construct, especially if you're trying to write with a mouse. Oh, let's use yellow. So what this is, what are the ratios that's going to be involved when we're done? It's going to produce a phenotypic ratio that is not typical to what we've seen before. And so what I'm going to do is let's color code this just a little bit. I like to do that a little bit. So let's say our first color coding is going to be in yellow, and this is going to be individuals who are going to have white fruit that is going to be disc shaped. Okay, and so that is a big W and a big D. So that's this individual, that one. So we're at 4, 5, 6, 7, 8, and 9. So we have 9 individuals who are going to have white fruit that is disc shaped. And let's go through. And we'll do magenta. Okay, we'll do magenta, and that is going to be white fruit that is not disc shaped, but it's going to be sphere shaped. So that'll be our lowercase d. So there's 1 individual, there's 2 individuals, and I'm going through 3 individuals, and that should be it. So there are 3 individuals that have white disc sphere shaped fruit. Okay, and then let's do blue. Okay, and blue is going to represent lowercase w, capital D. I hope that shows up well enough for you on the video. So lowercase, so that's going to be 1, 2, 3 individuals right here. So three of the individuals, or in this ratio, 3 out of the 16 will have yellow fruit that is disc shaped. And then finally, let's choose green. Hopefully it'll show up. This is going to be lowercase w, lowercase d, and there is just one individual that has that. Okay, so this ratio of 9 to 3 to 3 to 1 is typical of a dihybrid cross between individuals that are both heterozygous for both traits. Okay? So one of the most important things to remember when looking at probabilities is that when outcomes are independent, i.e. independent assortment, the probability of one outcome does not affect the other outcome. It's like tossing a coin. Okay? If you flip a coin, right, and it comes up heads, that doesn't mean the next time it's going to come up tails. Whatever happened before that is independent of what happens next. If you do enough coin flips, you should end up with a ratio that is 50-50. But you may need 1,000 coin flips or more to get to that point where it's 50-50, 50% heads, 50% tails. It's the same thing with rolling dice, right? If you roll, on a 6-sided dice, if you roll a 1 4 times in a row, what is the probability that you are going to roll a 1 the next time? Well, it's still 1 in 6, even though you rolled 4 before. You rolled 4 ones before that. It's still 1 in 6 each time you roll that dice. So the probability of independent events occurring together is equal to the product of those occurring separately. So the number of ways a given outcome can occur divided by the total possible outcomes is going to give us our probabilities again. It's still independent. Okay. All right. So I am not going to do this entire example with you because it will take me far too long and you guys are getting bored of listening to me right now. I know. This is just another example that would have you setting up a 4x4 Punnett square again with the 4 possibilities for each of the gametes from the man and from the woman. So the man and the woman will supply 4 different gametes: big E, big W, big E, little w, little e, big W, little e, little w. So they each supply 4. You'll set up the Punnett square like we did in the example just a couple slides ago, and you're going to end up with that 9 to 3 to 3 to 1 ratio. Okay, and 9 being dominant for the individual, 3 being dominant recessive, then recessive dominant, and then the 1 being recessive. Okay, I want to talk about one last thing here, and then I'm going to wrap it up. A polygenic trait is a single trait determined by more than one gene, and a lot of what we have in our bodies is polygenic. Poly, okay, meaning, and I'm sure you've probably spoken that out loud already. Poly meaning many, and genic referring to genes. So this is many genes determining a trait. So for us in humans, these are just some examples. Eye color is actually polygenic. And if you wanted to, you could look it up at some point and sort of figure out what it is. And I believe one of the Amoeba Sisters videos talks a little bit about polygenetic traits. Skin color is polygenic, height, intelligence, all of those things are polygenic, controlled by more than one gene. Genes that interfere with the expression of other genes are said to be epistatic. Again, the Amoeba Sisters talks a little bit about that as well. Situations that involve polygenic traits involve phenotypic and genotypic ratios that are significantly different from those provided by non-interacting genes. Okay, what that means is if we did a Punnett square analysis of eye color of a whole bunch of people, we wouldn't find a 9 to 3 to 3 to 1 ratio. We wouldn't find a 3 to 1 ratio. We wouldn't find any of those phenotypic ratios we're used to because it's controlled by more than one gene. It's controlled by 2 or more genes to give us the expression of our eye color. Okay, so it's important for you guys to understand what polygenic means and what implications that has on our expression, particularly phenotypic expression of genes. Okay, so that is finally the end of concept C5. Holy smokes. If you guys made it this far, good on you. This was a bit of a slog to get through this. 2 videos, probably close to an hour and a half worth of me chatting at you and trying to get this stuff done. So, like I said, hey, you made it through the longest concept in this unit. C5 is rough. When we get onto C6, 7, and 8, they're going to go fast. And so, you know, you'll have time to catch up. It won't be ridiculous. So don't panic too much if this took a lot out of you. At this point, you're good to go and move on to, I've got 3 more worksheets up there for you. At least try to get some of them done to get some practice with this stuff. So worksheet 2 has just some general questions like you might see on a quiz in there. So it's got some written out questions I'd like you guys to do. Please do worksheet 2. Have a look at, and it covers monohybrid crosses, so it'll go back and look at some of the stuff from the first part of C5, which is great. It'll do incomplete dominance, codominance, multiple alleles. Worksheet 3 has you working with dihybrid crosses. It's a really quick one. It's on guinea pigs. And then the last one is worksheet 4, which has some goofy worksheet I found somewhere that somebody made on Oompa Loompa genetics. So go and have a look at that one. If you've done all of that, checked your answers with the keys up in the Google Classroom, you can move on to the C4 and C5 practice quiz. Remember, once you've done the quiz, give me a chance to give you some feedback before you consider doing the summative quiz. So that is concept C5. Stay tuned for concept C6 and C7, where we're going to start to wrap up the rest of this unit. I wish you guys all the best in this. Track me down if you need help at any point. I am not far away. And as always, we will talk again soon.